Hey everybody, Mr. Grove here. Today we're going to talk about cellular respiration. And so cellular respiration is the way that your cells in your body gets energy from sugars. And so overall, this cycle is pretty interesting. So we have cellular respiration where your body is going to produce ATP made from chemical energy, so that's the sugars, and so that process, that reaction is glucose, which most foods are eventually broken down to glucose, plus oxygen, and then that reacts to make carbon dioxide, water, and then energy in the form of ATP. And so all living things do that, even plants actually, will do the process of cellular respiration. But then photosynthesis is kind of the opposite process of that. So photosynthesis is, the reaction is actually just backwards. So carbon dioxide plus water plus an energy source will turn into glucose or some other sugar plus oxygen. And so it's the same reaction, it's just arrows pointing this direction. And then if we flip the arrow, then we would get photosynthesis on the other side. And so it's basically a cycle. So the only thing that is not replenishable is the initial energy required for photosynthesis. And so that comes from the sun. And luckily, astronomers say we have a whole bunch of that left in the sun. So we're in pretty good shape. Another thing that we can kind of think of is the concept of heterotrophs and autotrophs. So heterotrophs are organisms that get energy from the food. They can't make their own. So they have to get their energy from the food that they eat. We are heterotrophs. Autotrophs are organisms that can use energy from the sun or other inorganic substances to make their food. So if we look at different things, so deer season is almost upon us. So a white-tailed deer would be a heterotroph. So they have to eat acorns and buds to get energy for themselves. Something like a plant here, okay, that would be an autotroph. So they can get their energy from the sun. A mushroom is going to be a heterotroph, so it has to break down things to get its energy. It does not have its own capability there. This red algae would be an autotroph, and so even though it's not green, it still has pigments that can do photosynthesis. This protist is a parasite, and so it's going to be a heterotroph. It has to get its own food from somewhere else. It can't make it. Our clownfish Nemo and his home the anemone, both also are getting their food, so they'd be heterotrophs. And then our green hydra, he's kind of actually an interesting thing. So he's a cousin of the jellyfish, and so he has to capture his own food, but there's some algae that lives symbiotically within him, and so that he has a little bit of photosynthetic ability as well. So we'll just kind of leave him halfway in the middle as kind of an organism that can do both. So heterotrophs get their energy, get their food from somewhere else. Autotrophs can make their own through photosynthesis. Okay, so the magic energy molecule is ATP. So that's adenosine triphosphate, and it has three components to that. So the base is what's called adenine, and then we have a sugar component, ribose, and then we have three phosphates, and that's where the triphosphate comes from. And so when we are spending ATP, we're actually releasing one of those phosphates, and so it's kind of like a spring that's been coiled up, and so energy gets released. And so that phosphate gets thrown off there, and energy is released when it does so. If we want to replenish the ATP or make ATP, then we have to stick that phosphate back on the ADP, diphosphate, so now we glued it back together, and now we have ATP again. But that requires energy for us to do that. So in the process of making the ATP, or using the ATP, then that gives off energy, or if we want to take ADP and make ATP out of it, then we have to supply energy. So the process that, where our cells get the ATP from is going to be called cellular respiration. And you definitely want to make sure you know this reaction, so know these 5 components of the reaction. So sugars plus oxygen gives carbon dioxide, water, and energy, and that is going to be our cellular respiration reaction. When things require oxygen, we call that aerobic. And if it does not require oxygen, we call that anaerobic. So when you're in gym class and you're doing jumping jacks, we call those aerobics because you start breathing hard and you need more oxygen. So some organisms do better without oxygen. Some organisms do better with oxygen. We happen to be one of those. And some can kind of do both. So of our stages of cellular respiration, let's go ahead and start with stage 1. And there are 3 main stages of cellular respiration. There's going to be one kind of minor, kind of intermediate stage that we'll talk about as well. But the 3 main steps or stages are going to be glycolysis, the Krebs cycle, and then the electron transport chain. And these are considered the main ones because they're all making ATP. So as you're filling out your student notes, you'll definitely want to make sure that you're taking into account how many ATP molecules are being made in each of these 3 main steps. So let's start off with glycolysis. So glycolysis, we need to make sure we know what goes in and then what comes out of this process. This is an enzyme-based process. And so it does not require oxygen, so it's anaerobic, and it takes place in the cytoplasm of the cell. And so as that glucose comes in, glucose is a 6-carbon molecule, and it's sometimes best to try and follow the carbons. So this 6-carbon molecule comes in, and we actually have to spend a little ATP to get things going. It's kind of like in the investment world. If you want to make money, you got to spend a little bit of money. And so the cell spins 2 ATP, and through a series of enzyme processes, we'll break that down into 2 3-carbon molecules. That produces what's called NADH. And so this NADH is kind of like a little electron taxicab that will take these high-energy electrons to a place later down the road. So we've created 2 NADH molecules. More enzyme process. And now we have ultimately created 4 ATP. So we spent 2, made 4, so our net total is 2. And then the end result molecularly is going to be these 2 molecules called pyruvate. So we got 1, 2 pyruvate molecules, and those are 3 carbons each. So what we started going into the process here, 1 glucose, and then coming out was 2 pyruvates, so 6 carbons, 2 3 carbons. We made 2 ATPs ultimately, and so to do that we had to bring in then 2 ADPs, and if we filled up 2 electron taxicabs called NADH, then that means the empty taxicab molecules called NAD+ had to go into the process. So on your student notes, the ins and the outs, what goes in? 1 glucose, 2 ADP, 2 NAD+, and then what comes out of this grooming or this glycolysis process it would be 2 ATPs, 2 NADHs, and 2 pyruvates. So that is glycolysis. Our next step is the grooming process, and this one isn't considered a main step, probably because we're not making ATP out of it, but it's still important. And so in the grooming process that takes place in the mitochondria, we have our 2 pyruvate molecules that are coming in, the ones that we made in glycolysis. And so those pyruvate molecules are going to come in, and through a series of enzymes, we are going to cut 1 carbon off. So what started with 3 carbons is now going to end up with 2. So where's the carbon go? Carbon dioxide. And to do that, we have to have this thing called coenzyme A. So coenzyme A will come in and attach to those 2 carbons, and that will make a molecule called acetyl-CoA. So pyruvate comes in, cuts a carbon off, we add coenzyme A, and then we end up with acetyl-CoA. This whole thing gives us some electrons that can fill up one of our little electron taxicabs, NAD+, and that forms an NADH. And since we had 2 pyruvates going into this process, we get to do this twice. So ultimately, coming out of the process would be 2 acetyl-CoA molecules, 1, 2. Coming in would be 2 pyruvate molecules. And since we made 2 NADHs coming out, that means coming in had to be 2 empty NAD pluses. And then the carbon dioxide comes out, and there was 2 of those as well. So that's the grooming process. And that takes place in the mitochondria. And we're grooming the next step, is basically what that means. The next step is going to be the Krebs cycle. Sometimes it's called the citric acid cycle, but you can call it either one. It's also enzyme related, and it takes place in the mitochondria. So we kind of think of these as the aerobic stages because they're in the mitochondria now. And so what's going to come in are those 2 acetyl-CoA molecules that we made from the grooming process. And so that acetyl-CoA comes in, and it joins up with a 4-carbon molecule that was already there. And so that 4-carbon molecule called oxaloacetate then joins up, and we end up with a 6-carbon molecule, and that is called citric acid. And so that's kind of where the citric acid name comes from. Another enzyme cuts off a carbon. So if we cut off a carbon, now we're down to 5. And so that carbon can be released as a carbon dioxide. That enzyme process fills up an NADH. If we go from 5 to 4 carbons through another enzyme, that means we get another carbon dioxide, and that process makes an ATP and fills up another NADH. Our enzyme next, another enzyme will take this 4-carbon and rearrange it to this 4-carbon. And that process fills up more electrons. This time the taxi is a little different. It's what's called FADH2. Okay, so the empty version would be FAD, the full version would be FADH2. It does the same job as NADH, it's just a little less flashy. It doesn't have the high energy. So where this would be more like a limousine taxi, this is like a minivan. Still going to get you where you need to go, but not as fancy. Next, after that 4-carbon compound is going to use some enzymes to go back to our oxaloacetate, and that will be another NADH that we're filling up. And so we originally get back to our oxaloacetate. And so we're ready for another acetyl-CoA to come in. And so the cycle will just turn again. And ultimately then what comes out is 2 ATP, 1 for each turn, so 1 here and 1 on the next turn, 6 NADHs, 3 on each turn, 2 FADH2s, 4 carbon dioxides, 2 on each turn, and then 2 coenzyme A's. So after the acetyl-CoA glues those 2 carbons to the oxaloacetate, we get 6. That coenzyme A goes back to the grooming process to get more carbons. And so that's what comes out. So going into the Krebs cycle is going to be 2 acetyl-CoAs, 1 for each turn of it, 6 empty versions of NAD+, to pick up those electrons and make NADH, 2 empty versions of FAD, and then the 2 ADPs that we made our 2 ATPs out of. And so that is the Krebs cycle. Now finally, then, to this point, we've only made 4 ATP, and we have to get to about 40 for the cell to be efficient. So we have a whole lot of ATP left to make, and we have a whole lot of NADH that we have made. And so the last step is going to be the electron transport chain. And so this is what we would consider an aerobic step, still taking place in the mitochondria, and this is where the majority of our ATP is made. So somewhere in the 34-ish range, 34 to 36 range. So we have to make a lot of ATP, and so let's get after it because it's got to be done. So all of these electron taxi cabs are showing up. It's kind of like the prom. You know, all these limousines are showing up, dropping the kids off, and so they show up here at the electron transport chain. And this takes place in the cristae of the mitochondria, so this mitochondrial lining. And so it drops the electrons off, and those electrons then jump from protein to protein. Okay, this first protein is called phosphofructokinase, and it picks that protein up, and then it passes it to the next protein, passes it to the next protein, and so on and so forth. What causes it to pass? Well, that would be oxygen. So oxygen is very electronegative. It's kind of like an electron magnet, and it just pulls those electrons from here to here to here to here to here, and then eventually they end up in the oxygen. Every time an electron jumps from protein to protein, we're going to pass hydrogen ions across the membrane of the mitochondria. And so the outside of this has a whole lot of hydrogen ions, and the inside not as many. And so we have a concentration gradient, a difference. And so down here there's an opening, and this is what's called ATP synthase. And this is the magic, this is where the magic happens. So those hydrogens will go back through the ATP synthase, and that's kind of like water going over a dam or a water wheel, creating energy. And then that gives it the energy to glue this phosphorus to an ADP molecule, and that's what makes our ATP. Okay, so as we pump hydrogens across to this side, they come back through this only opening, and that's where the energy can be made to make the ATP. What happened to the electrons that were doing the jumping? Well, they ended up getting grabbed by this oxygen here, and then these hydrogens ions also can get grabbed by the oxygen. And so if we have hydrogens, electrons, and oxygens, that basically is water. And so that water is going to be also a byproduct of the electron transport chain. So NADH coming in, dropping off all the electrons, FADH2 dropping off a few electrons, oxygen has to be part of it, and then we have to have the uncharged ADP part. And then what comes out is the charged up ATP, water, and then the empty limousines, the empty minivans that don't have their electrons anymore will go back to the other steps, glycolysis and Krebs, to pick those up. And so this whole process ends up making, like we said, about 34 to 36 ATP. So the majority of your ATP is made in this electron transport chain. So in review, we have our 3 steps: glycolysis, the Krebs or citric acid cycle, and then the electron transport chain. And we were going to be making 2 ATP, and about 34 to 36 ATP for a total of about 38 to 40-ish ATP. And that's pretty good. We still had more that we could have done, but your body just can't make it as efficient to do that. And so that is how cellular respiration works when it's working right. However, there's a couple times where we don't want things to work right. If we can somehow mess up the cellular respiration of an organism, that might cause that organism to die. And sometimes that's what we're after. So maybe that's a weed in a field, maybe that's an invasive species bacteria, maybe that's an insect like the emerald ash borer beetle, the Asian carp. If we could figure out how to disrupt their cellular respiration, we could maybe make that organism die and bring the ecosystem back to its balance. So here are some common poisons that you've kind of heard of, some of them, and we'll show you how they work in the electron transport chain to make that not work. So the first one was rotenone. And so rotenone was a chemical back in the 70s that was very common for an insecticide. And so basically what it did is it blocked the electron transport chain right here at the start, that first protein. So the electrons had a traffic jam. They had nowhere to go. If we're not moving electrons from here to here to here to here, then that means we have no hydrogens being pumped across. If we don't have a hydrogen concentration gradient, meaning more on this side and less on this side, there's no reason for hydrogens to go through the ATP synthase. And if they're not going through the ATP synthase, they're not making ATP. And so no ATP means the organism is going to starve of energy and end up dying. 2 other ones that you have heard of, cyanide and carbon monoxide. So if you watch the spy shows, they're always using the cyanide tablets. And then carbon monoxide this time of year is a very dangerous poison in homes. And so what carbon monoxide and cyanide does is it blocks right here before the oxygen. So if that electron can't get to the oxygen, then that means this electron can't get to its place. So that means there's no electrons to get to his place. And so even though the traffic jam is further up the road here than what rotenone was, it still creates the same effect, a traffic jam. And so the traffic jam means no hydrogens are pumped across, no hydrogens want to go through the ATP synthase, and therefore no ATP is made. Another type is oligomycin. And oligomycin is an over-the-counter antifungal cream, so it's used for like ringworm and things like that. Oligomycin, what it does is it just blocks the ATP synthase right here. So we're still doing everything. We're creating all these hydrogens over on this side. They want to get to this side, they just can't. So if they can't go through, no ATP, means no energy in the organism, will die. One last poison is the uncouplers, and so they are a group that belongs to things like dinitrophenol. This was like a dieting weight loss drug at one time. And so what it did is it created a leaky mitochondria. So it would allow everything to kind of work right. So we didn't have a block here. We didn't have a block here. Those electrons went ahead and jumped. We produced hydrogens on this side. And so those hydrogens, some of them would go through. We would make some ATP. But then since there's holes, it's a leaky mitochondria, there's holes in the lining, so some of the hydrogens just went through the membrane. Well, that's not going to make any ATP. It only can make ATP through the ATP synthase. So as it's going through, we're making some ATP, but not enough. And so we're slowly starving the cell, which for a weight loss thing is good. You're slowly starving it. And so you'd have to rely on fat reserves and you'd lose weight. This worked well, but the problem with this is dinitrophenol turned out to be a carcinogen and potentially cancer-causing, and so that's how it got banned from the pharmaceuticals then. But these are just kind of 5 poisons and how we can disrupt the cellular respiration cycle, specifically here the electron transport chain, and that will allow the organism to not succeed. So that's cellular respiration, both good and bad, and I hope that was helpful.